Hello and welcome to our presentation. Today we will be discussing the article B-Cell Memory, Building Two Walls of Protection Against Pathogens. To start off, we will take ourselves all the way back to 430 BCE, where a plague in Athens ran rampant through the city. Amidst this plague, survivors began to recognize their immunity, bravely caring for the infected, knowing that, in their words, no one was attacked twice, at least not fatally. This stood as the basis for Edward Jenner's groundbreaking development of the smallpox vaccine in the 18th century that induced lifelong immunity. However, as modern science advances, the quest for effective vaccines still faces challenges. We will journey into the depths of immune memory of long-lived plasma cells and memory B cells that play a vital role in the long-lived protection against pathogens. Originating from the bone marrow, B cells undergo maturation, emerging as defenders that can recognize a variety of pathogens. Upon encountering an antigen, B cells can differentiate into two distinct lineages, long-lived plasma cells and memory B cells. Long-lived plasma cells remain in the bone marrow and produce an abundance of antibodies specific to encountered antigens. Memory B cells circulate through the body looking for pathogens they have encountered before to initiate a rapid response. Together, these B cell subsets form a strong defense network, ensuring the body has both immediate protection and long-term immunity against threats. Next, we will be looking deeper at the two phases of B cell memory.
Phase 1 of B cell memory is characterized with the antigen-driven differentiation of naive B cells. Follicular dendritic cells present the antigen to naive B cell receptors. Naive B cells then activate and express chemoattractant receptors that direct the B cell to the border of the T cell zone. At the border of the T cell zone, the activated B cell interacts with antigen-specific T follicular helper cells, which provide multiple helper signals to guide B cell differentiation. Through CD40L, CD40, and ICAM1 LFA1 pathways, and the secretion of IL4 and IL21. Next, the activated B cells undergo proliferation and follow one of three different fates. Naive B cells can differentiate into short-lived plasma cells that produce antigen-specific, low-affinity antibodies during the span of infection, or GCB cells that undergo somatic hypermutation in germinal centers, or GC-independent memory B cells with limited isotope switching. Now you may be wondering, what determines which cell type that the naive B cell will differentiate into? One study used mice models with rare naive B cells expressing human germline VRC01 class BCRs, and found that the precursor frequency of naive B cells, antigen affinity, and avidity were interdependently limiting for successful GC completion. Another study suggests that the weak expression of interferon regulatory factor 4 tends to drive GC B cell differentiation, while high levels of IRF4 expression promote plasma cell differentiation. The third study showed that through CD40-CD40L signaling, T follicular helper cells direct naive B cells to differentiate into GC B cells in early stages of infection, but would shift to favor plasma cell differentiation in cases of chronic CD40 signaling. Interestingly enough, though, antigen affinity of naive B cell receptors has little effect in the first phase of differentiation. A study found that despite naive B cells receptors having much lower antigen affinity than GC B cells, this does not prevent naive B cells from entering germinal centers and differentiate into high-affinity plasma cells and B cells.
In this section, we will look at the formation of high-affinity B cells in germinal centers. First, GC B cells proliferate and undergo somatic hypermutation in the GC dark zone. These cells then enter the GC light zone to differentiate into long-lived plasma cells that constantly secrete antibodies, or long-lived memory B cells that are ready to encounter new infection. If necessary, GC B cells can also undergo further somatic hypermutation and affinity selection upon new infection to differentiate into either long-lived plasma cells or memory B cells. GC B cells sense their affinity to antigens through the following processes. First, B cell receptors, or BCRs, are attached to the antigen-presenting membrane of follicular dendritic cells, forming a BCR antigen complex. Second, myosin, a motor protein inside B cells, pulls the BCR antigen complex to deform the antigen-presenting membrane. Lastly, B cells internalize the BCR antigen complex and are ready to present to T helper cells. The pulling forces of myosin allow GC B cell to test antigen affinity of cell surface receptors. High-affinity GCB cells can form stable and long-lived binding with antigen-presenting cells and demonstrate more efficient antigen internalization and presentation. Meanwhile, these high-affinity GCB cells activate both CD40 signaling pathway through the nuclear factor kappa B and BCR signaling pathway through the FOXO1 protein, which collectively induce the key transcriptional factor protein MYC to ensure cell survival and outcompete low-affinity GCB cells. This allows the selection of only high-affinity GCB cells, which have 100-fold higher antigen affinity than naive B cells, and induce long-term survival and memory.
Instruction include protein kinase C-beta and intersectin 2. One study highlights that protein kinase C-beta deficient mice cannot develop plasma cells or germinal centers following immunization, due to its role in regulating B-cell polarization and metabolic programming. Another study discovered that a deficiency of intersectin 2, a guanine exchange factor used for cytoskeleton remodeling, undermined the formation of B-cell T-cell conjugation. This means that an abundance of intersectin 2 is essential for B-cell T-cell interactions and further B-cell differentiation, which in turn impacts the level of protection after immunization. Antigen structure also plays a role in how much protection immunization confers. One such factor includes the presence of weakly immunogenic recombinant subunit antigen, which are still capable of producing a strong antibody response, as evidenced by the highly effective HPV vaccine. Another component is how the antigens are delivered in vivo. The two modes of delivery include soluble delivery, in the case of small antigens, and membrane-associated delivery for larger antigens. Although the fate of responding B cells has yet to be studied, it is understood that both types of antigen delivery require different parameters for successful B cell activation. The follicle microenvironment has also been implicated in B cell development. One factor is the presence of antigen or pathogen-associated molecular motifs, or PAMPs, within the B cell follicles. Researchers discovered that CpG ligand for toll-like receptor 9, a PAMP, prevents naive B cells from internalizing antigen or being co-stimulated by T cells, which drive B cell differentiation into short-lived plasma cells producing low-affinity antibodies. This was a surprising result, given how adjuvants like CpG ligand for TLR9 are routinely used to further stimulate the immune system during immunization, and it raises questions as to what adjuvants can be used for future immunization strategies to better optimize B cell response.
In this section, the B cells are destined to evolve into either long-lived plasma cells or memory B cells. So what determines whether GC B cells become either one? Studies have shown that the GC B cells tend to form memory B cells in the early stages of infection and gradually shift to long-lived plasma cells later on. The differentiation trajectory is influenced by several key factors. The initial phase primarily favors the generation of memory B cells with relatively low rates of somatic hypermutation, thus providing broad, a little bit less potent antigenetic coverage. In contrast, there is a temporal shift in plasma cell production after GC B cells maturation, with long-lived plasma cells exhibiting a higher degree of antigen affinity and potency to provide an immediate effective defense. There are functionally distinct subpopulations of memory B cells compartment, each with unique phenotypic markers and poised to play specific roles upon reactivation, allowing for nuanced responses to changing pathogens. Understanding these mechanisms of differentiation and selection is critical to vaccine development because it provides insight into how to elicit a balanced immune response that produces both potent high-affinity antibodies by long-lived plasma cells and a broad, adaptive pool of memory B cells in response to change pathogens.
While vaccination has been pretty effective thus far, we've been having a lot of trouble making vaccines for big problems like AIDS and malaria. So, What do we do if we can't generate a long-lived memory B cell response? A way we've been circumventing this issue is through prophylactic antibodies. And the transfer of antibodies to provide preemptive protection is something we've been doing for quite a while. For example, with hepatitis B and hepatitis C. However, this protection is only temporary and needs fairly frequent injections of antibodies to ensure protection. Recently, though, researchers have been looking into vector mediated antibody gene transfer, which will provide long lasting protection. Looking to the future, protective monoclonal antibodies and antivirals are in clinical development, and the emerging field of vector mediated antibody gene transfer has shown some promise. Between these, we'll hopefully find some good ways to generate protection against AIDS, malaria, and other tough pathogens. So, today we've learned how B cell memory develops, what it does, and our limitations with manipulating it. Further studies in long term protection generated by B cells might be useful in learning to make the most out of our innate immune response.